Hello students, welcome back to the biology class. So in the last class we learned about pedigree analysis. We understood that it is a graphical representation or it's a family tree which represents the record of the inheritance of any character. It can be genetic disease, it can be a genetic good character from a trait and its inheritance for two or more generations. Males in this whole concept are called as propositae and the females are called as proposita. We understood that the experiments which were conducted by Mendel and Morgan on their selected organism Pisum sativum and Drosophila melanogaster, those experiments are not possible to be conducted on the human because it is unethical first of all. Second point, number of chromosomes are large. Many of the human characters are polygenic and they are influenced by the environment. And then marriage occurs in the case of human in different families. Homozygosity is very rare in human, and then there are small number of offsprings formed, but the generation time is long, and an observer or an experiment performing person can maximum see only 2 to 3 or 4 generations at a time. Because lifespan of human also is quite small. So that's why pedigree is the only option left. It's a tool for understanding inheritance, and this helps us to understand the probability of diseases to be transferred from generation to the next generation, and thus it is used by counselors to advise couples, newly-wed couples. If they have a history of some genetic disorder throughout the family, then they are counseled by the counselor not to go for kids, because if there is higher probability of having a disease carrying kids, then they will become a burden on their parents also. So such counseling things are all done with the help of pedigree analysis. And then we learned that there are certain symbols for pedigree. A box is for the male, a round one for the female, and if it is of this shape, the sex is not specified. If it is filled, affected individual. This line represents mating but distant family, close relatives. This line represents that next generation is coming up. This is the order of the kids born. The left side is the first one, right side is the next one. A number in the box represents the number of individuals. This type of bracket around represents adoption. P means pregnancy, miscarriage, death, no offsprings, double marriage, one left or then the other one second marriage. Now this is identical twins. If line is not there, non-identical twins. Roman numbers are for generation. The Arabic digits are for the individuals in the generation, like first individual, second individual. And then this represents the carrier. Generally this is used, these two are used generally. And this is the stillbirth. So we did some of the pedigrees in the last class. Just wanted to remind you once again, Y-linked characters will only be visible in the male, but all the males in all generations. So they pass on from father to the son to the grandson. And autosomal dominant will definitely not skip any generation, and autosomal recessive will skip generation. Then sex-linked dominant will be more visible in female, but sex-linked recessive is more visible in the case of male, and will follow the crisscross inheritance. Remember crisscross, from one sex to the opposite sex, then come back to the same sex. That is called as crisscross inheritance. Now the next topic to start with is the last topic of this chapter, and with this we are closing this chapter, and that is the human genetic disorders. So now we are going to understand many types of the disorders which are common in the human. Remember, we have learned already that the disorders are of how many types? Just a second. We learned that these mutations are chromosomal mutation or gene mutation, and chromosomal mutations can be structural change or the number change, and gene mutations are also possible. So we are going to learn today this whole concept, and we will taking some diseases out and their symptom and their cause to finish this topic now. So chromosomal disorders. Now, chromosomal disorder. First one, the easiest one, can be the genomatic or the number disorder. So, if the number of chromosomes change, then also diseases are possible. We have learned in mutation already. Now, there are two types of chromosome in human: autosomal chromosomes, or you can say the non-sex chromosome and the sex chromosome. So, two types of disorders are possible: a disorder because of autosome number change, or the allosome number change. So the first one to study is the Down syndrome. Okay, let's do one thing. Let's read quickly from the NCERT first, then we will be doing it from the notes. So NCERT says chromosomal disorder. This topic, chromosomal disorder, here means the number of the chromosome. So chromosome disorders, on the other hand, are caused due to the absence or the excess or the abnormal arrangement of one or more chromosome. Failure of segregation, this is the answer. Failure of the segregation of chromatids during cell division cycle result in the gain or loss of the chromosome, and this is called as aneuploidy. For example, during Down syndrome, result of the why Down syndrome happen? Because there is a gain of an extra copy of which chromosome? 21st chromosome. So you have to learn this number. The 21st chromosome extra copy is the reason behind Down syndrome. Similarly, Turner syndrome is because of loss of an X chromosome. So the female will have just one X and the other X is finished or it's not there. So XO. Failure of the cytokinesis. Now this is different. So reason of aneuploidy is this and reason of polyploidy is this. Now the polyploidy reason,